Hello. Very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible, and also if you can see my video clearly. Let me know if everything is fine, and if you can see the PPT clearly. Okay, thank you so much. We will wait till it is the time, and till more students join, then we will start today's session. Hello. So am I audible? There was a minor problem for the internet connection. Please let me know if I am audible. Okay, thank you so much. Okay, so it's 6 p.m. I will wait for some time more, maybe or 2 to 3 minutes more, and otherwise I will start the session. Okay, so it's 6 p.m. Let's wait for a few more time and then we can start the session. Meanwhile, I hope my voice is audible to you and you can see my video and the PPT clearly. If not, please let me know in the chat box. And very good evening to the students who have joined. We will wait for 2 more minutes and then we will start the session. Let me know if there is any problem with the voice or audio or the presentation or the video. Anything, if there is any problem, just let me know. Okay. Okay, I think it's better if I start with the introductions. So, very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry and today it is our week 10 live session. So, in this week 10 live session today, it is our 10th live session and every week we just take a week of that which week is live on that per day, and we solve around 20 MCQs regarding related to the topics of the week which is live right now. So, right now the week 11 is live and today we will discuss 20 questions related to the week 11 course content. Okay. Although these questions will be MCQs, but after showing you the answer, or after having a after checking if you can answer that question correctly or not, I will discuss that question thoroughly, and in this way I will discuss the topics of the course along with. Okay. So like every week, today we will also have this kind of live session in Google Meet, and just please let me know if there is any problem with the voice, audio or video, anything. So the chat box is open, okay, and anything, if you want to tell me anything, any related to the topics or the class, you can just let me know in the chat box. Okay. For the I am recording today's session because I have to upload it later, so for the recording purpose I have muted you all and kept the cameras off so that I can record it very smoothly. Okay. But if you have any doubt, any query, the chat box is always open and I am monitoring the chat box. So if you have anything you want to tell me, you can just let me know in the chat box. So before starting the session, I will just clarify a few more things which I do before every class. So there will be a problem solving session every week, okay, till your course is over. So your course, it has a 12 week, it is a 12-week course, right? So we will have 12 problem solving session, among which today it is the 10th one, okay. And apart from the 12 problem solving session every week, on Tuesday 6 p.m. to 8 p.m. we will also have a 1 hour summary session. The date and time of the summary session will be communicated to you very shortly via email, okay. What will be the in that summary class, I will summarize the whole course, and also in the last problem solving session, which is our 12th problem solving session, there also I will summarize the course, and we will have this few question answer sessions from overall the course, okay, and along with the topics which I couldn't discuss very broadly, but which is very important for the medicinal chemistry. That things I will discuss in the summary session. So please do join the summary sessions. The schedule will be discussed to you soon. These sessions will be live on the Google Meet platform, and the Google Meet platform link is same for every week, okay. You can just save this Google Meet link, and you can use it for joining on every class on Tuesdays. If any network disruption happens for my side, please do not leave the class, okay. Do not leave the classroom, and please wait for few minutes. I will join as soon as possible. But if there is any network issues from your side, you can rejoin at any time, okay. The session is open, okay. You don't need to take permission every time. Just you can join at any time during the session if there is any network issue from your side. So during the session, the students can post their question, their doubts, any query related to the course or any topic, okay. Not only for today's week class, you can just drop any your question or queries in the chat box about the previous classes also, okay. But still, if any doubt persists after class, I will share my email ID at the end of the session. So you can just drop an email to me, where I will discuss or I will answer your question or your query in the next class, okay. And this presentation and the recording, which I am recording the session, so this presentation and the class recording will be shared to you soon. Okay, so I think it is time. I will just start the session. When most students will join, they can just join at any time. If you have any problem, let me know in the chat box, okay. It is always open. So as I have said earlier, today it is our week 10 live session, and today what we will discuss is, we will discuss the topics which are in your week 11. So let's see first what are the topics of week 11. So the topics are, so these are very interesting topics of medicinal chemistry. When I was reading medicinal chemistry during my MSc or PhD time, so these topics are very important and also very interesting. So we have today, what we have is like antiviral agents, which are divided into part 1 and part 2. Then we have anti-cancer agents, we have cholinergics and anti-ulcer agents. So let's see what are the question of today's session. And I know all of you know how we do in this kind of sessions. I will show you one question, and I will give you some time, maybe a time of 3 to 4 minutes, and you will have 4 options, A, B, C or D. Just you have to answer correctly in the chat box, okay. A, B, C or D, which of the following you think is the correct one, you have to let me know. And after checking the answer, after 3 to 4 minutes, I will show you what is the correct answer, and we will discuss that question thoroughly, okay. So let us start with today's first question. So the first question of today's class is, which of the following statements is true regarding virus? Your options are: number A, viruses are not living organisms; number B, viruses grow and reproduce outside the host cells; number C, tuberculosis is a viral disease; and number D, viruses are giant compared to bacteria. So which of the following statements is true regarding virus? I have told you earlier also in earlier previous sessions, whenever we are having this kind of true false question, that these questions are very, sometimes this question can be tricky. So read carefully all the options and then let me know A, B, C or D, which of the following is the correct answer. Even if your answer is wrong, but please answer, okay, otherwise I won't understand if you are following the class or not, or you are if you are understanding the things or not. Okay, so let me know A, B, C or D, which of the following is true regarding virus. Option number A, viruses are not living organisms. Number B, viruses grow and reproduce outside the host cells. Number C, tuberculosis is a viral disease. And number D, viruses are giant compared to bacteria. Which of the following statements is true? Take some time, I will give you 2 to 3 minutes and then I will check the chat box and we will discuss it. Which of the following statements is true regarding the virus? Okay, let me check the chat box and then I will discuss it. Okay, so there are options number A. So all of you think it is the number A. Okay, let me check if number A is the correct one or not. So the correct answer will be yes, number A. Very good. Viruses are not living organisms. So let us see. So what is a virus? So definition is that the virus is a pathogen or disease causing agent not considered living because it cannot reproduce on its own. Okay, so this is, there is a structure of the virus. So we will discuss about these structures of virus in later in our later slides in detail. So there are two different things are like one is virus and one is bacteria. These two common pathogens are very common because we know that there are whatever diseases we have right now today, it is very common that we are either having a bacterial infection or a viral infection, which are more common. Okay, so what are the differences between the virus and bacteria? So viruses are not living organism, whereas the bacteria are considered as living organism. Viruses only grow and reproduce inside the host cell they infect, and when found outside the living cell, viruses are dormant. Their life therefore requires the hijacking the biochemical activities of a living cell. Bacteria are living organisms, and they are that consist of a single cell, can generate energy, makes its own food, move, reproduce, typically by binary fission, and this allows bacteria to live in the many places like soil, water, plants and human body, and serve many purposes. Viruses are submicroscopic, bacteria are giant compared to the viruses. A viral infection is systematic. Viruses infect a host cell and then multiply by thousands, leaving the host cell and infecting other cells of the body. Bacterial infection is usually confined to the part of a body described as a localized infection. Infections may cause by the bacteria or by toxins like endotoxins produced. Systematic diseases caused by viral infection include influenza, measles, polio, AIDS and COVID-19. And bacterial infections include pneumonia, tuberculosis, tetanus and food poisoning, etc. So, these are very common, are very important differences between the common pathogens, bacteria and virus. Okay. So, option number A was the correct one. So, now I will move toward the next problem. At any point of the session, if you think there is any problem of understanding of that answer, please let me know in the chat box. Okay. So, now problem number 2, it says that identify the correct order of viral infection in the host cell. Your options are number A, attachment, followed by uncoating, followed by penetration, then release, then assembly and then biosynthesis. Option B is attachment, followed by penetration, then uncoating, then biosynthesis, then release and then assembly. Number C, attachment, followed by biosynthesis, then penetration, then uncoating, then assembly and then release. And number D, attachment, followed by penetration, then uncoating, then biosynthesis, then assembly and then release. So, what is the correct order of viral infection in the host cell? Look at all the options very carefully and then let me know A, B, C or D, which of the following is the correct one? Which of the following is the correct order of viral infection in the host cell? Attachment, uncoating, penetration, release, assembly, biosynthesis, option number A. Option number B, attachment, penetration, uncoating, biosynthesis, release and assembly. Number C, attachment, biosynthesis, penetration, uncoating, assembly and release. And number D, attachment, penetration, uncoating, biosynthesis, assembly and release. Which of the following will be the correct one? Okay, let me check the chat box now. Okay, I have few answers. Number D, number B, number D, number D. Okay, so there is a little confusion about number B and number D, right? Okay. Now let me check which of the following is the correct one and then we will discuss it. So, the correct answer, the correct order of viral infection in the host cell will be number D, attachment followed by penetration, then uncoating, then biosynthesis, then assembly and then release. Now, let me show you. So, what are the steps of the viral infection? Most viruses follow the similar series of steps in the viral reproduction cycle. These are attachment, penetration, uncoating, replication, assembly and release. Once inside a host cell, a viral DNA or RNA is released and can begin to interact with internal cell machinery. Viral DNA can direct transcription of viral mRNA, which then translated into the viral proteins. Viral RNA can be used as a template to make more viral mRNA and proteins. RNA retrovirus use reverse retranscription to make viral DNA, which is then transcribed and translated. Regardless of the mechanism, the objective is the same: more virions. New virions are released when the damage to the host cell causes lysis, or when the host cell undergoes apoptosis, which is the programmed cell death. Some viruses are released through the budding, which releases one virion at a time and spares the host cell. So, again these are the steps. So, this is the nucleus and this is a host cell, okay, and this is a, let us say it is a, for example here it is the influenza virus which is shown here. So, the first step is the attachment. So, what is the attachment step? So, in this step this virus becomes attached to the target epithelial cell, okay, and then the second step is the penetration, where the cell engulfs the virus via endocytosis. After that there is a uncoating. Uncoating means the viral contents are now released into the cell cytoplasm. Then is the biosynthesis. So, in the biosynthesis step, viral RNA enters the nucleus, where it replicated by the viral RNA polymerase, and then we have the assembly. Assembly is new phase new Fudge particles are assembled, and then the release. New viral particles are made and released into the extracellular fluid. The cell which is not killed in the process continues to make new virus. So, this is a virus infected cell. At first it is in one cell is infected, and when new viruses are made and released, the process is the in this process this cell is not damaged, okay. It will continue to make new viruses that is the process of the viral infection of the host cell. So, I think it is now clear that the steps are attachment, penetration, uncoating, replication or biosynthesis, assembly and then release. Okay. So, now I will go to the next problem. So, problem number 3, it says that which of the following statements is false regarding the characteristics of a good protein target for antiviral drugs? The options are: number A, it should be important to the life cycle of the virus; number B, it should bear little resemblance to human proteins; number C, it should be important in the late stages of the virus life cycle; and number D, it should be common to the different types of virus. Which of the following statement is false regarding the characteristic of a good protein target for antiviral drugs? Number A, it should be important to the life cycle of the virus. Number B, it should bear little resemblance to human proteins. Number C, it should be important to the late stages of virus life cycle, and number D, it should be common to different types of virus. Which of the following statements is false regarding the characteristic of good protein target for antiviral drugs? Okay, let me check the chat box now. Okay, I have few answers, number A, number D, number C, number D, number C. So I have 3 different answers for this question, number A, number C and number D. So let me check which of the following is the correct one, and then let us discuss. So the statement which is false regarding the characteristic of the good protein target for antiviral drugs is number C. It should be important in the late stages of virus life cycle. This statement is false. Now let me show you the types of the antiviral agents and the key characteristics. So antiviral agents can be of broadly 2 types. First is the targeting viral function, and the second is targeting the cellular function of that virus needs. Okay, and the targeting viral function can be like the targeting these things like the nucleic acid polymerase, protease, integrase and neuraminidase. Okay, so then the nucleic acid polymerase can be of 3 type: DNA dependent DNA polymerase, RNA dependent RNA polymerase and DNA dependent RNA polymerase. So all these are the antiviral agents or the targets which can be the enzymes or the which can be targeted for the antiviral agents, okay, for making antiviral agents. Now the key characteristic of antiviral drugs: they should able to enter the cells infected with virus, interfere with viral nucleic acid synthesis or regulation. Some drugs interfere with the ability of virus to bind to the cells. Some drugs stimulate the body's immune system. Best response to antiviral drugs are with patients with competent immune system. A healthy immune system works synergistically with the drug to eliminate or suppress the viral activity. Okay, so this is so the statements that it should be important to life cycle of virus, it is correct. It should bear little resemblance to human protein, it is also correct, because if the particular target or the protein target resembles too much with the human proteins, so what will happen, there won't be any selectivity. Okay, it should be important in the late stages of virus life cycle, it is not necessary, because it should be important to the every step or early steps of virus life cycle, otherwise it won't be very much useful. It should be common to different types of virus, it is also correct, because otherwise we will have to take lots of medicine for different viral disease. Okay, so the option number C is the false regarding the good protein target of antiviral drugs. Okay, so now I will move toward the next problem. Problem number 4 says the HIV protease enzyme is symmetrical, whereas mammalian proteases are not. What significance might this have? Your options are: number A, inhibitors of HIV protease enzyme must also be symmetrical. Number B, it may be possible to design inhibitors that prove selective for HIV protease over mammalian protease. Number C, only symmetrical substrates are cleaved by the HIV protease. And number D, there is no significance. The HIV protease enzyme is symmetrical, whereas mammalian proteases are not. What significance might this have? Your options are: number A, inhibitors of HIV protease enzyme must also be symmetrical. Number B, it may be possible to design inhibitors that prove selective for the HIV protease over mammalian protease. Number C, only symmetrical substrates are cleaved by HIV protease. And number D, there is no significance. So which of the following A, B, C or D will be the correct one? Let me know in the chat box. Read the options very carefully and then let me know. let me check the chat box now. Okay, if you have any doubt, any query, you can let me know in the chat box. Okay, so the options, I think you have answered number B. All of you think it is a number B. Okay, let me check if it is the correct one or not. So very good, number B is the correct one. It may be possible to design inhibitors that can prove selective for the HIV protease over mammalian protease. So the HIV protease enzyme is a very good and very important target for antiviral drug designs, which for the HIV disease. Okay, the HIV protease enzyme is symmetrical dimer made up of two identical protein units, each consisting of 99 amino acids. The active site is at the interface between the protein units and is also symmetrical with twofold or C2 symmetry. The enzyme has a broad substrate specificity and can cleave a variety of peptide bonds in the viral polypeptides, but crucially it can cleave bonds between a proline residue and an aromatic residue, phenylalanine or tyrosine. Moreover, the symmetrical nature of the viral enzyme and its active site is not present in mammalian proteases, again suggesting the possibility of drug selectivity. Okay. So, this is the reason, this is the significance of the human HIV protease structure, which is different from the viral protease, sorry, the mammalian proteases. Okay. So, now problem number 5, it says that steroid hormones combine with intracellular receptors to form substrate that control the expression of genes. What are these structures called? The options are number A, nuclear translation factors, number B, nuclear transcription factors, number C, nuclear replication factors, and number D, nuclear reproduction factors. I will again repeat, steroid hormones combine with intracellular receptors to form structures that control the expression of the genes. What are these structures called? Number A, nuclear translation factors, number B, nuclear transcription factors, number C, nuclear replication factors, and number D, nuclear reproduction factors. Okay, let me check the chat box now. Okay, so the options are given are number C, number B and number B. Okay, let me check which of the following is the correct one. All of you please try to answer, okay. Even if your answer is wrong, it doesn't matter, so please participate. So the correct answer will be number B, nuclear transcription factors. So what are the transcription factors of eukaryotic cells? So activator proteins bind to the pieces of DNA called enhancers. Okay, so there are these are the this portion of the DNA is known as the enhancer, and these pink this portion are the activator proteins. Okay, their binding causes the DNA to bend and bringing them near the gene promoter, even though they may be thousands of base pairs away. Okay, and these are other transcription factor and proteins, and these are now other transcription factor proteins, which are these ones, join the activator proteins forming a protein complex which bind to the gene promoter region, and this is the promoter, the gene promoter region. This protein complex makes it easier for RNA polymerase to attach to the promoter and start transcribing a gene. This is the RNA polymerase, and then an insulator can stop the enhancers. This is the insulator. These insulators can stop the enhancer from binding to the promoter if a protein called STCF, okay, and this which occurs to the insulator, named for the sequence CCCTC, which occurs to all insulators, binds to it. This is the insulator, okay, the CTCF, and then methylation, the addition of a methyl group to the C nucleotides prevent the CTCF from attaching to the insulator, turning it off, allowing the enhancer to bind to the promoter. Okay, so these are the steps, and these are the for the transcription factors of the eukaryotic cells. Okay, and so again I will just repeat the steps. First, the activator proteins bind to the pieces of DNA called enhancers. Their binding causes DNA to bend and bringing near the gene promoter region, and there will be other transcription proteins which will form a protein complex which binds to the gene promoter. Here is a gene promoter. That protein complex make it easier to RNA polymerase to attach to the promoter and start transcription, and there will be an insulator which is known as CTCF or CCCTC binding factor, which can stop the enhancer from binding to the promoter if the protein binds to it, and methylation, the addition of the methyl group to the C nucleotides prevent the CTCF from attaching to the insulator, turning it off, allowing the enhancer to bind to the promoter. Okay, so nuclear transcription factor was the correct answer. So now I will go to the next problem. Problem number 6 says smallpox and yellow fever are serious viral infection, but these can be tackled by, your options are number A, antiviral drugs, number B, natural product, number C, vaccination, and number D, immunotherapy. Smallpox and yellow fever are serious viral infection, but these can be tackled by antiviral drugs, natural product, vaccination, or immunotherapy. A, B, C or D, which of the following is the correct one? Let me know in the chat box. All of you try to answer the questions. I will wait for 2 to 3 minutes, and then I will check and let you know what is the correct answer. Okay, let me check the chat box now. Okay, number C, number C, number C, number C. Okay, all of you think it is a number C. Okay, let me check if it is the correct one. So the correct answer will be, yes, it is the number C, the vaccination. So one ways is in which we can protect ourselves from the viral disease is to use vaccination. A number of diseases we looked, such as polio, measles, mumps, are actually mainly targeted by vaccination. So smallpox, yellow fever are also sort of serious disease which can be countered by the vaccination. So what is a vaccination? Vaccination works by introducing the body to foreign material which bears molecular similarity to some component of the virus but which lacks the infectious nature or toxic effects. The body then has the opportunity to recognize the molecular fingerprint of the virus, the specific antigens, that is the specific antigens, and the immune system is primed to attack the virus. It should it infect the body. Vaccination, usually a killed or weakened version of the virus is administered so that it does not lead to infection itself. Alternatively, fragments of the virus, subunit vaccination can be used if they display a characteristic antigen. Vaccination is a preventive approach and is not usually effective on patients who have already become infected. Okay, so vaccination was the correct one. So now I go to the next problem. Okay, before that I will just show you one more thing about the vaccination. So the first, this is the your virus, right? So there are several structures. I have earlier told you that I will discuss a few about the structures of the your virus. So there is a spike glycoprotein at the outer face of the virus, okay, and these spike proteins are used to attach to the particular your host cell. This virus spike protein has a few portions, like there is a S1 subunit of the upper portion, then is a S2 subunit the lower portion, and we have a RBD, and then we have a membrane protein or which is M protein, envelope protein, E protein, nucleocapsid, N protein, and genomic ssRNA. And spike glycoprotein I have told you earlier. So first, there is a inactivated vaccine based on the chemically or physically inactivated virus grown in the cell culture. Okay, live attenuated vaccine which utilizes a weakened version or the limited replication extent of the virus in living cell. Viral vector vaccine that adapts existing successful and safe modified viral vectors to express coronavirus proteins. DNA vaccine contains the circular DNA that encodes the protein of the interest. RNA vaccine uses mRNA packed in a lipid nanoparticles, okay. Then we have recombinant subunit vaccine composed of the synthetic nanoparticles coated with the purified viral antigen of the spike protein, and virus like pyrogen particle vaccine contains a multiprotein structure that mimic the virus yet devoid of the viral genome. So these are the different types of vaccines which take uses the various component of a virus viral proteins and various viral structures, and then it uses and utilizes various portions. So these are the different kind of vaccines which can be used. Okay, so now I will move to the next problem. Problem number 7 says the coronavirus binds to human epithelial cells to dash receptor. Your options are number A ACE2, number B ACE1, number C BCR-ABL, and number D AT1. So which of the following receptors are responsible for the binding of coronavirus to the human epithelial cells? Number A ACE2, number B ACE1, number C BCR-ABL, and number D AT1. All of you try to answer the question and let me know in the chat box A, B, C or D which of the following you think is the correct one, or which of the following is the correct receptor for the binding of coronavirus to human epithelial cells. Okay, let me check the chat box now. Okay, number B, number A, number B, number A, number B, number B, number B. Okay, so number B and number A, these are the two answers, and there is a confusion about either it is number B or number A. Okay, now let me check if it is number B or it is number A, and then we will discuss about it. Okay. Okay. So the correct answer is number A. Okay. Okay, sorry, there is a confusion. I have just put it otherwise. It is number A, AC2. Yeah, it is correct. So number A, AC2 is the correct answer. Okay. So now I will show you about the coronavirus, which is a very important, I think, important phase of all of our lives. Okay. So first there is I will just go to this diagram, and then I will just go to the replication cycle. So let's look at the diagram. Okay. So very important thing is the spike protein of the coronavirus. The spike protein genetic material is extracted, and this is the how the coronavirus vaccine is made. Okay, the coronavirus vaccine is made. This is how. So this I will just tell you this later. First, let us understand what is the replication cycle of coronavirus. Okay. So infection begins when the viral spike protein is glycoprotein attached to its complementary host cell receptor. After attachment, a protease of the host cell cleaves and activates the receptor attached spike protein. Depending on the host cell protease available, cleavage and activation allows the virus to enter to the host cell by endocytosis or direct fusion of the viral envelope with the host membrane. On entry into the host cell, the virus particle is uncoated and its genome enters the cell cytoplasm. The coronavirus RNA genome has 5' methylated cap and a 3' polyadenylated tail, which allows the RNA to attach to the host cell's ribosome for translation. The host ribosome translates the initial overlapping open reading frame of the virus genome and forms a long polyprotein. The polyprotein has its own protease which cleave the polyprotein into the multiple non-structural proteins. A number of the non-structural proteins coalesce and to form multiprotein replicase transcriptase complex or RTC. The main replicase transcription protein is the RNA dependent RNA polymerase. It is directly involved in the replication and transcription of the RNA from RNA strand. The replicated positive sense genomic RNA becomes the genome of the progeny viruses, and these viruses are then released to the host cell by exocytosis through secretory vesicles. Infected carriers are able to shed viruses to the environment. The interaction of the coronavirus spike protein with complementary cell receptor is central in determining the tissue tropism, infectivity and species range of the released virus. Coronavirus mainly target epithelial cells. They are transmitted from one host to another depending on the coronavirus species by either an aerosol, formite or fecal route. Human coronavirus infect epithelial cells of the respiratory tract, while animal coronavirus generally infect the epithelial cell of the digestive tract. SARS coronavirus, for example, infects an aerosol route, human epithelial cells of the lungs by binding to angiotensin converting 2 enzyme receptor. We all know angiotensin converting 2. This enzyme is a very crucial enzyme in case of the for renin-angiotensin system, which controls the blood pressure of our body. Okay, so AC2 receptors are the one which are the where the coronavirus binds, because it is in the respiratory tract and the epithelial cell of the lungs. That's why we were told to keep the masks on all the time, so that this our respiratory tracts can be protected from the spike protein of the coronaviruses. So this is the spike protein. I have already told you. So how the coronavirus these vaccines are made. Okay, the spike protein genetic material at first are extracted and genetic material inserted into the inactivate harmless virus. Okay, this unrelated harmless vector virus is this, and then viral vector vaccine is made. Okay, so this harmless virus enters to the cell at first, the human cell. Then COVID-19 virus spike protein is created. Okay, and then antibodies of this particular COVID, this is the COVID-19 virus spike protein type. This is acts as an antigen, and this for this antigen antibodies are made, which are the spike proteins are recognized by immune system, which produces a specific antibodies against the COVID-19 virus. And this if you are infected with the COVID-19 virus, antibodies bind to the virus and stop them for replicating. So this is the way the COVID-19 vaccines are made. Okay. First, a portion of viral spike proteins are extracted and added to a harmless vector. Then it is added to the body. So for that, the viral spike protein antigens are formed, which will rise to the antibody. And if you are infected with COVID-19, so what will happen? This antibody will stop from the replicating. Okay. So now I will move to the next problem. Problem number 8, it says, which of the following enzymes is inhibited by acyclovir? Your options are: number A, viral protease; number B, viral RNA polymerase; number C, viral DNA polymerase; and number D, viral spike protein. Which of the following enzymes is inhibited by acyclovir? Viral protease, viral RNA polymerase, viral DNA polymerase, or viral spike protein? I will wait for 2 more minutes and then I will show you the answer and we will discuss it. Okay, let me check the chat box and then let us discuss about it. number C, number D, number C, number A, number A. A, C and D. I have 3 different options as answer. Okay, let me check which of the following is the correct one. So, the correct answer is C, viral DNA polymerase. This enzyme is inhibited by acyclovir. So, acyclovir, first I will just show you. So, acyclovir, then first it is a herpes specific thymidine kinase, and then by this herpes specific thymidine kinase, acyclovir is converted to acyclovir monophosphate. Okay, this acyclovir is the prodrug. First it is converted to monophosphate, then by this action of the cellular kinase it is converted to acyclovir triphosphate. Okay, this acyclovir triphosphate get incorporated in the viral DNA and stop the lengthening the DNA strand. The terminated DNA inhibits the DNA polymerase irreversibly and inhibit the herpes virus DNA polymerase competitively. Okay, so the DNA polymerase is the enzyme which is inhibited by acyclovir. First, the phosphorylation reaction that occurs by this enzyme is known as thymidine kinase, and here the enzyme is present in the host as well as the herpes virus, but it turns out to be selectively towards the herpes viral. Thymidine kinase is 100-fold higher. So, therefore, the thymidine kinase which is present in the virus is actually more effective in doing phosphorylation, so compared to the host cell, and therefore, this is the reason it is selective. And also there is selective uptake of acyclovir in the infected cell. Together this explains the excellent activity and reduced toxicity for the previous drugs developed in this area. Another feature is shows a 54 selective action against viral DNA polymerase relative to the cellular polymerase. So, this drug is one of the most important drugs developed against the herpes virus. Okay. And this acyclovir is the conversion of acyclovir to acyclovir monophosphate is the step which is the, we can say it is the activation from the product to active drug of this particular this but this prodrug to the active form of the drug, this transformation is catalyzed mainly by the herpes specific thymidine kinase, not the host cell thymidine kinase. Okay, that is why infected cells are mostly uptakes this acyclovir very well, and then it is more selective toward the viral DNA polymerase in comparison to other cellular polymerases. Okay, so viral DNA polymerase is the correct answer. So, now problem number 9, it says which of the following statements is true regarding HIV protease enzyme. Your options are: number A, it belongs to the enzyme family of cysteine protease; number B, it catalyzes the formation of peptide bonds; number C, active structure contains cysteine moiety; and number D, active site structure contains aspartic moiety. Which of the following statement is true regarding HIV protease enzyme? Again I will repeat, options are: number A, it belongs to enzyme family of cysteine protease; number B, it catalyzes the formation of peptide bonds; number C, active site structure contains cysteine moiety; and number D, active site structure contains aspartic acid. Which of the following statement is true regarding HIV protease enzyme? Read all the options very carefully and all of you try to answer the questions, since there is no marks given, but answering will help you to understand the things better, and also it will give you the confidence for understanding these topics. Okay, I will give you 2 minutes, think and let me know in the chat box. After 2 to 3 minutes I will check and I will discuss it. Okay, let me check the chat box now. Number D, number D, number D, number D. Okay, so all of you think it is the number D. Okay, let me check if number D is the correct one. So the statement which is true regarding HIV protease enzyme is, yes, it is number D. Active site contains aspartic acid. So the function of HIV protease, one long chain of HIV proteins inside the infected cell. This is the protease enzyme. What does it do? It cuts the particular long chain to two shorter chains of HIV protease and individual proteins that make new HIV particles, and individual enzyme that can bind to help to new particles. Okay, the protease inhibitor is which stops this individual proteins which makes new HIV particles. So this is the structure of the active site, where we have the aspartic acid or aspartate residues. Okay, and this is how the particular function happens. You don't need to remember all these mechanisms, just I have given it for your understanding. So first we are having two aspartate residues. Okay, aspartate as 225 and 25 prime, and then we have a water molecule which is hydrogen bonded with these two aspartate residues. Okay, and we have this our chain of the peptide bond, and then what happens, this particular water oxygen of water, it acts as a nucleophile, and we have this kind of tetrahedral intermediate. Okay, and this tetrahedral intermediate is what which cleaves this peptide bond, and we have this two different parts. Okay, and this is the that's how this protease cuts this HIV long chain, and this cut into the two half. Okay, and then there is the regeneration of the enzyme by working of the similar way. Okay, there is several important examples for the HIV protease inhibitors like indinavir, nelfinavir, ritonavir, saquanavir, ampiramir. All are these examples of the HIV protease inhibitors, which are very important drugs. Okay. So now I will move to the next problem. Problem number 10 says the following structure of saquanavir is a protease inhibitor. This decahydroisoquinoline ring, okay, the decahydroisoquinoline ring means this one. System in the protease inhibitor was used to replace an amino acid residue which is present in the lead compound. Which amino acid was replaced? Your options are number A phenylalanine, number B aspartic acid, number C proline, and number D tryptophan. Again I will repeat, this is the structure of saquanavir, which is a protease inhibitor. This decahydroisoquinoline ring system was used to replace an amino acid residue which was present in the lead compound. Okay, what amino acid was replaced? Number A phenylalanine, number B aspartic acid, number C proline, and number D tryptophan. All of you try to answer the question in the chat box. I will give you 2 minutes and then I will check. let me check the chat box. Okay, number C, number A, number D, number B. Okay, so I have different answer. Number A, B, C and D. I have 4 different options all in the chat box for the first time, I think, which is very good that all of you are thinking in different ways. Okay, let me check A, B, C or D, which of the following is the correct one. So the correct option will be number C, proline. Proline is the amino acid which was replaced by this decahydroisoquinoline ring. So saquanavir is sold under the brand names like Invirase and Fortovaz. This binds to the active site of viral protease and prevents the cleavage of the viral polyproteins. Okay, this is a little bit tricky, this different portion of the, this is the basically this is around the SAR of the particular group, medicine particular drug. So first there are different components, as you can see. Okay, this ligand means this was the different things which were in the first, it was something different in the your lead compound, and then it was changed. Okay, so first is the orange one, which is this portion, as you can see this one. This is the P1 ligand. This is unsubstituted benzyl group is preferred in this position. Larger groups, for example pyridyl, methyl, piperidine, moiety, this also interact with the S3 site. Okay, so then we have this this blue portion, which is this chain portion. So this is a P1 prime peptide bond transition state binder. The OH group is crucial. This OH group is crucial to mimic the catalytic transition state, forming key hydrogen bonds with carboxylic acid group and aspartate and sp25 and 25 prime. Hydroxylethylamine, hydroxyethylene, hydroxypentane, azathiohydroxyne are usually used in transition mimic moieties. Then we have this black one, which is the Pa2 prime ligand. Different hydrophobic, alkyl and aromatic groups are tolerated here. Cis amino hydroxylidine moiety in IDV led to 100-fold increase in potency compared to the benzylamine analog. Thiazolyl moiety in RTV increases the solubility and benzyl sulfonamide derivatives bearing the substituents in para position for amine, OMe, OH are able to form hydrogen bonds, are optimal for potency. Then this red one is the P1 prime ligand to decrease the peptide character. Okay. So this P1 probe has been replaced with the bicyclic decahydroisoquinoline SQV in other please different hydrophobic groups. In other protease inhibitor, different hydrophobic groups have been used like phenyl tertiary leucine, isobutyl. Use of the sulfonamide and carbamate to link the P1 ligand improves the pharmacokinetic properties, including better oral bioavailability. Then we have this P2 ligand. ASN residue was replaced both to reduce the peptide character and increase the binding affinity. Carbamate and uridio linker increase the metabolic stability. Good ASN mimics the thiazolone moiety in RTV, cyclic urea unit in LPV, and tertiary butyrate in IDV. And then we have this P3 ligand, which is the greenish one. Large hydrophobic groups can accommodate in this unit in this following generation of your protease inhibitors. P3 ligand has been removed because not necessary for activity of interaction outside the envelope. So these are different portions or different groups. You don't need to remember each and everything. Just read this thing whenever I will share you this PPT. Read this thing. I think you will understand how this particular changes can happen. Okay. And if you don't understand, just let me know. I will discuss in the next class also this thing. Okay, or into the summary class. Now I will go to the next problem. Problem number 11 says the following structure belongs to a group of compounds obtained from the African bush willow and shows promise as an anti-cancer agent. Which of the statements is not true? Number A, above structure is being tested as phosphate prodrug in cancer chemotherapy. Number B, it prevents angiogenesis. Number C, the cis-trans isomers are equally effective. And number D, it bears structural similarity to colchicine. Which of the following statement is not true regarding this particular anti-cancer agent? Okay, now let me check the chat box. Number D, number A, number B. Okay, I have different answers again. Okay, let me check which of the following is the correct one. So the correct answer will be number C. The cis trans isomers are equally effective. This statement is false. Okay. So this is basically known as combristatin. It is a very important anti-cancer agent, which is the tubulin polymerization inhibitor. Okay. So there is a microtubule, there are tubulin alpha and beta from this microtubule polymerization happens, and which gives to the angiogenesis and okay. So what does this combristatin do? It has an antiproliferative activity. It inhibits the microtubule formation, and which inhibits the cell cycle of the cancer cell, and inhibits the cell migration also, and again this blocking of this microtubule polymerization leads to the BCL2 activation, which gives rise to the apoptosis of the cell, which known as the cell shrinkage, chromosomal and condensation, membrane blobbing, nuclear collapses, and apoptotic body formation. This combristatin as a cis structure is the more effective than the trans structure. Okay, the trans structure is not as much effective as the cis structure of combristatin. So statement number C was the false. Now problem number 12, which says that the cell growth checkpoint is present in dash phase of the cell cycle. Your options are number A M, number B G1, number C S, and number D G2. Cell growth checkpoint is present in which phase of cell cycle? M, G1, S or G2? Let me check the chat box now. S. Okay, S phase. Number A, number B, number D. Number A, B, and number A. Okay. So I have answers as option number A, option number B, and somebody else says it is the number S, which is the S phase. Okay. So let me check. So the cell growth checkpoint is present in number B, G1 phase of the cell cycle. So this is the phases of cell cycle. We have different phases of your cell cycle. The following describes the what different checkpoints of cell cycle are responsible for. First we have a G1 phase, which is the, and in this G1 phase, then we have a S phase, which is the and then we have G2 phase and M phase. So G1 phase, in G1 phase is known as the growth phase 1. Okay, and at the end of the growth phase 1 or G1, we have a cell growth checkpoint. In the S phase, this checks whether the cell is big enough and has made of the proper proteins for the synthesis phase. If not, the cell goes to the resting period, which is a Z0 phase, until it is ready to divide. Then we have a DNA synthesis checkpoint in S phase, which is known as synthesis phase. It occurs during the synthesis phase and checks whether the DNA has been replicated correctly. If so, the cell continues to mitosis. Then we have a G2 phase, which is another growth phase, and then last we have the mitosis checkpoint in the M phase or mitosis phase, where it checks whether mitosis is complete, and if so, the cell divides and the cycle repeats. Cancer can result when one or more of these checkpoints malfunction. Okay. So now next problem, problem number, okay, this is 12. I think there is a problem in number. Okay, just forget the number, it is the problem number 13. Okay, kinase inhibitors must bind to the dash region of the enzyme. What is the correct answer? Number A hinge, number B N lobe, number C C lobe, and number D DFG region. Kinase inhibitors must bind to which region of the enzyme? Hinge, N lobe, C lobe, or DFG? Let me check the chat box. I think there is a problem in the number of the questions. Okay, it was number not 15, it was number 13. Okay, so the answers are number A, number C, and I think it is for the previous ones. Okay, let me check which of the following is the correct one. So the correct answer will be number A, hinge region. So basically kinases are the enzymes which, what does it do? It takes up the phosphate, the terminal phosphate from the ATP, and it adds to a particular protein target. Okay, this target protein must have a OH group. Okay, so the amino acids which have terminal OH are serine, threonine, and tyrosine. So basically kinase enzymes are three type, either serine, threonine, or serine threonine kinase, or tyrosine kinase. This terminal OH group is phosphorylated by this from the phosphate group from ATP, and the protein kinase, it catalyzes this transfer of phosphate group from ATP to this protein target, and this phosphorylated protein, then what it will do, it will create some cell signal which are responsible for various works of our bodies. Okay, but due to this cell signaling of the kinase enzymes, different in cancer cells, these are very are crucial. Okay, some cell signaling, they actually related to the cancer. So kinase inhibitor are the compounds which inhibit the work of the kinase enzyme selectively, so that the particular the cell signaling which is responsible for the proliferation of the cancer cells, it does not happen. So what is this? Let us focus on the structure of the kinase. So this is a one example of how the kinase enzyme looks. So first we have this is the upper portion, it is called the N lobe. So what this is the N terminal lobe. Okay, and here we mostly have the beta pleated sheets, and alpha helices are present mainly in the lower portion, which is a C terminal lobe. Okay, and there we have this a particular portion of a protein which joins this N lobe with a C lobe. It is known as the hinge protein. Most of the 90 to 95% of kinases have the similar structure, and which is very easy to find out what is the hinge region. When you just look at the structure, you have to just check what is the N lobe, what is the C lobe, and the hinge binding at the connection between the N lobe and C lobe. Okay, and there we also have a particular the residue, which is the first amino acid residue of the hinge region, is known as the gatekeeper residue. Okay, and then we have a particular, in this place we have 3 amino acids all together, which is D, F, and G, which is known as the DFG loop. So these are the all kinase inhibitors which are till now these which are approved by FDA for say various cancers. Okay, you don't need to just remember this. You just I have just given it for your understanding. So these are different kinases like ROCK kinase, EGFR, HER kinase, BCR, ABL, mTOR, VEGFR, RET kinase, JAK kinase, all are different kinase enzymes, and these are the compounds which inhibit this particular, and this is the year where the kinase inhibitors are approved for every year. So most of the kinase enzymes they are so this is a suffix for remembering, like if it is a it is tyrosine kinase, and this is all this are given here, the nomenclature portion, and most of the kinase inhibitors are approved for the cancers only, and there are few kinase inhibitors which are approved for different other diseases also, but they are not very much. Okay, like your ruxolitinib, it is I think for human arthritis. Okay, so these are the examples of kinase inhibitors which are approved as drug. So now problem number 14, it says that palbociclib is an inhibitor of dash kinase. Your options are number A JAK, number B BCR ABL, number C CDK4/6, and number D MET kinase. Palbociclib is inhibitor of which kind of kinase? JAK kinase, BCR ABL, CDK4/6, or MET kinase. I will give you some time, think, and then let me know. Let me check the chat box now. Number C, number C, number C. So all of you think it is a number C. Okay, let me check if number C is the correct one or not. So the correct answer will be number C, CDK4/6. Okay. So, cell cycle inhibition by CDK4/6 inhibitors. So, first I have shown you earlier also G1, S, G2 and M. We have this 4 phases in cell. So, this particular G1 cell accumulates resources that need to duplicate the genome. In S, actual duplication of genome occur, or that is a DNA synthesis. Then G2, proper DNA replication checked and final preparation of mitosis phase and mitosis phase. Chromosomes are separated and actual cell division occurred. And CDK4/6 inhibitors, what does they do? So, basically they inhibit in this acts on this portion, okay, and this is the quiescence scale, which is the cell starts, okay. So, palbociclib is the structure of is this, it is primary target is CDK4 and 6. So, type 1, it is a type 1 kinase inhibitor, binds to the ATP pocket in an active conformation through 2 hydrogen bond with kinase hinge, valine 101 and 1 with aspartate 163 of DFG moted. Its cyclopentyl group occupies the ribose pocket, this one. Treatment of hormone receptor HR positive, HER2 negative advanced or metastasis breast cancer in post-menstrual women. The side effect is neutropenia and leukopenia. Okay. So, now next problem number 15, it says that imatinib is an important anti-cancer drug which targets a protein kinase. What role does the piperazine ring has? This is the piperazine ring, which is the blue one. Number A, it increases water solubility. Number B, it increases target selectivity. Number C, it increases target binding. Number D, all of the above. Imatinib is an important anti-cancer drug which targets protein kinase. What role does the piperazine ring has? It increases water solubility, it increases target selectivity, it increases target binding, or all of the above. Okay, let me check the chat box now. Number D, number A, number C, number A, number A. Okay. So, I have different answer again. Let me check. So, the correct answer will be number D, all of the above. This piperazine ring has the role that it increases water solubility, target selectivity and also the target binding. So imatinib is a very important BCR-ABL kinase inhibitor. So how does it work? Basically, this is the BCR-ABL kinase. Okay, we have this ATP which converts the ATP and PI. This is the substrate binding pocket, and this substrate is converted to phosphorylated ones and then effector and signaling. So what does the imatinib do? Imatinib mesylate, it binds to the ATP binding portion of this substrate of this BCR-ABL kinase, and now ATP cannot bind and phosphorylation can occur and cell signaling cannot occur. So there is a different amino acids which binds with the different portion of imatinib, like methionine 319 with this pyridine ring. Okay, and this this pyrimidine ring which is present, there is a NH group which is binded with the threonine 315, and glutamine 286 binds with this NH, like aspartate 381 binds with this O, and histidine 361 and isoleucine 360 binds with the piperazine ring. This portion of the imatinib binds to the adenine pocket of kinase, this to the hydrophobic pocket, and this piperazine portion to the allosteric pocket. And this is the protein structure which is I have shown you earlier also. This is the hinge portion. Okay, and this is the imatinib which binds with this portion here via various interactions. So now I will go to the next problem. Problem number 16 says anti-ulcer drug omeprazole is number A reversible proton pump inhibitor, number B antagonist of H plus K plus ATPase, number C agonist of H plus K plus ATPase, and number D irreversible proton pump inhibitor. Anti-ulcer drug omeprazole is which kind of drug? Number A reversible proton pump inhibitor, number B antagonist of H plus K plus ATPase, number C agonist of H plus K plus ATPase, and number D irreversible proton pump inhibitor. Let me check the chat box now. Number B, number D, number D, number D, number B. So I think there are number B and number D. The confusion arises from number B or number D. Okay, let me check which of the following is the correct one, number B or number D. So anti-ulcer drug omeprazole is number D, irreversible proton pump inhibitor. So this is the structure of omeprazole. So what is the proton pump inhibitors? Proton pump inhibitors are a group of drugs which main action is pronounced and long lasting reduction of gastric acid production. So omeprazole, lansoprazole inhibit the protein pump effectively and irreversibly, requiring synthesis of new enzyme protein. Omeprazole and lansoprazole approved for the treatment of duodenal ulcer, erosive esophagesis and gastric acid hypersecretory states including Zerzinger-Ellison syndrome, which gastronomas that cause secretion of large amounts of acid. Okay, so proton pump inhibitor, omeprazole is the proton pump inhibitor. So now next question. Problem number 17 says again about it is about omeprazole. Omeprazole is a proton pump inhibitor. Okay, which region is prone to metabolism? This is the structure of omeprazole. We have this benzimidazole ring, then we have this pyridine ring, and we have a methyl group here, OMe group here. Okay, all these things are here. So there are 4 regions are mentioned here. One is this blue methyl, then this brownish O methoxy, then we have this C methyl red one, and this D phenyl ring. So which of the following region is prone to metabolism? A, B, C or D? Which of the following region is prone to metabolism? Let me check the chat box. Number A, B, A, D. So I think A, B and D are the 3 different answers which are given. Okay, let me check. So the correct answer is number A. Okay. So basically this omeprazole, okay, so in case of omeprazole, so omeprazole, there is a in blood the pH is 7.4, in cytosol the pH is 7.1, and secretory glands it has the pH of 2.0. So omeprazole first it inserts to the omeprazole inserts to the cytosol, and then it converts to the ATPase and H plus kinase, and there are other functions also here are mentioned. This representation is also better. Okay, so there is a receptor, histamine H2 receptor, various M3 receptor, and also we have a CC2 receptor present here. Okay, and this all is the there is a ATP, H plus ATPase, NPS, and this is the gastric gland or lumen. Okay, and omeprazole, first there is omeprazole in blood. Okay, the structure is this, which is given also here. So when this omeprazole which is present in blood, it goes to the parental cell of lumen. Okay, what happens? This particular, there are 3 steps by which it the metabolization happen. Okay. So first step is the there is a proton, insertion of proton, this particular ring, this pyridine ring is protonated, and first step. Then in 3 step what happen? In 3 step this kind of sulfonamide intermediate is formed. Okay, and then we have H plus K plus ATPase. Okay, and this H plus A plus ATPase, it binds with this portion, and we have enzyme inhibitor complex. Okay, so this A portion is more prone to the metabolism otherwise, because it is just next to the particular position. Okay, so this position is most prone to the metabolism. So now problem number 18, it says that method of action of aromatase inhibitor is, your options are number A irreversible inhibition, number B reversible inhibition, number C suicide inhibition, and number D uncompetitive inhibition. Method of action of aromatase inhibitor is irreversible inhibition, reversible inhibition, suicide inhibition, uncompetitive inhibition. So what is the method of inhibition of aromatase inhibitor? Irreversible, reversible, suicide inhibition or uncompetitive inhibition. Let me check the chat box now. Number B, number B, number D, number D, number D. So I think I have number D and number B are most given answer. Okay. Let me check if the number B or number D is the correct. So correct answer will be number C, suicide inhibition. So the method of action of aromatase inhibitor is suicide inhibition. So what does the aromatase inhibitor do? First let us know what is the aromatase. So it is an enzyme that catalyzes the conversion of androstenedione or testosterone to estrone and estradiol. Okay, and this aromatase this estrone and estradiol, first estrone is related to the ER activation and proliferation of mutated cells, and estradiol generation of E2 344 quinone, and then ER activation, proliferation of mutated cell, and DNA alkylation or depletion, which leads to the cancer. So aromatase inhibitors which inhibits this aromatase. So first I will show you what is the this is the mechanism of the active site mechanism. The first we have this aromatase, okay, and then we have this 4-member porphyrin ring with iron 2 and a cytosine. Okay, then what happens, we have this oxygen which makes this pyramidal structure to a square planar structure, and then we have this oxygen as a radical, this hydroperoxide radical first we have, which converted to the anion, which attacks on the particular this structure of estrone, and then what we have is there is a protein, there is a proton, and this kind of active site mechanism happens. So you don't need to just remember all this thing, just remember it is a kind of the suicide inhibition what this aromatase inhibitors do. Okay, and what is the suicide inhibition and all this type of inhibition I already have discussed earlier when I was discussing about the enzyme kinetics, and in the summary session also at last I will summarize all these things. Okay, what is the different kind of inhibition in case you have forget. So now next problem number 19 says, which of the following activities occurs following the stimulation of H2 receptors? Your options are number A, vasodilation, number B, uterine contraction, number C, bronchial smooth muscle contraction, and number D, enhanced secretion of hydrochloric acid in stomach. Which of the following activities occur following the stimulation of H2 receptors? Vasodilation, uterine contraction, bronchial smooth muscle contraction, or enhanced secretion of hydrochloric acid in stomach. Let me check the chat box now. Okay, so number D is the given answers. Let me check if number D is the correct one or not. So the correct answer will be, yes, it is number D. Enhanced secretion of hydrochloric acid in the stomach is the activities which occurs during the stimulation of H2 receptors. So histamine receptors, there are different histamine receptor, H1, H2, H3 and most recently H4. These are G protein coupled receptor. Activation of H1 cause the contraction of smooth muscle in gut, uterus and bronchi. Contraction of bronchi leads to asthma. Stimulation of H1 receptors in smooth muscles in fine blood vessels cause muscle relaxation and resulting vasodilation may result in several fall in blood pressure. In CNS, H1, H2 receptors predominantly localized on the postsynaptic membrane. H3 receptors appear to function predominantly as presynaptic receptors, possibly in histamine autoreceptors, and also be detective in some peripheral organs. H4 receptor exhibit very restricted location in intestinal tissues, spleen, immune active cells, T cells, suggesting therapeutic potential of H4 blocker in allergic and inflammatory diseases. The differences between H1 and H2 receptor, H1 receptor, histamine receptors that couple with GQ11 stimulating phospholipase C, and these receptors that interact with GS and activate aniline cyclase. Periphery of the body, adrenal medulla, vascular endothelium, heart and CNS is the location for this H1 receptor, and CNS and periodontal cells located in the stomach is the location of H2 receptor. Antagonist diphenylamine, loratadine, cetirizine are all these are the antagonist of H1 receptor, and ranitidine, cimetidine, these are the antagonist of H2 receptor. Function, ileum contraction, modular circadian cycle, itching, systemic vasodilation, bronchial contraction are the function of H1 receptor, and speed up the sinus rhythms, stimulation of gastric acid secretion, smooth muscle relaxation, inhibit antibody synthesis, T cell proliferation, cytokine production are the function of the H2 receptors. Okay. So now I will go to the last question of today's session. It says, what sort of receptor is muscarinic receptors? Your options are: number A, G protein coupled receptor; number B, kinase link receptor; number C, intracellular receptor; and number D, ion channel. What sort of receptor is muscarinic receptor? G protein coupled receptor, kinase link receptor, intracellular receptor, or ion channel? What sort of receptor is muscarinic receptor? Let me check the chat box. Number A. Okay, I think number A is the given answer. Okay, since we don't have much time left for today's class, let me just check if number A is the correct one or not. So the correct answer will be, yes, number A, G protein coupled receptor. So first, let us understand how G protein coupled receptors work. So these receptors are the largest class of membrane protein in eukaryotes. They mediate most of the physiological response of hormone, neurotransmitter and environmental stimulants. When a G protein coupled receptor is not bound to agonist, it remains inactive, and G protein also remains inactive in cell membrane. Upon binding the ligand or agonist such as hormones and neurotransmitters, the G protein coupled receptor undergoes conformation change, activates the GEF domain. The GEF domain activates the monomeric GTPase to replace the GTP in the GS alpha domain, activating the G protein. Upon activation, the GS alpha domain dissociates from the GPCR protein couple and binds to the effector enzyme on the cell membrane to activate it. The activated effector enzyme generates the second messengers that activates the intracellular signal transduction cascades. The hydrolysis of GTP into GDP and GS alpha domain dissociates the GS alpha domain from effector enzyme, deactivating the enzyme. and muscarinic acetylcholine receptors are classified from different M1 to M5. Here I have discussed only M1, M2 and M3. The M1 in CNS, salivary glands and parietal cells, which is responsible for CNS excitation, memory, locomotor activity and gastric acid secretion. M2 in heart, which drops the rate, force and AV contraction, and smooth muscle and exocrine is M3, all smooth muscle except contraction, vasodilation and glandular secretion. Okay, M4 and M5 are mainly the present within the CNS, and they have exact but the exact role are unknown. Okay. So, this was the last question of today's session. So, today it was the week 10 live session for our course, where we discussed the topics of the week 11, okay, which is live right now. I hope this kind of questions will help you to understand this week 11 topics more better, and also it will help you for answering your weekly assignment. Okay. So, today what we discussed, we started with the antiviral agents. We discussed a lot about antiviral agent, the targets of the antiviral agents and related to this. Then we move forward to the anti-cancer agents like kinase inhibitors. Then we discussed few about the cell cycle, how it works, and also the about combristatin, which is a tubulin polymerization inhibitor. And lastly, we discussed about the cholinergics and proton pump inhibitors. So, now I will share my email ID with you in the chat box. Okay. So, this is my email ID. If you have any doubt, any query regarding today's session or any topic, from any topic, since we are almost at the last of our this kind of problem solving sessions, we will have 2 more problem solving sessions, week 11 and week 12, and 1 summary session of 1 hour. Okay. So, and then you have your examinations. So, if you have any problem, any doubt from any portion of the course of medicinal chemistry, you can let me know in the email. Okay. In the last 2 classes and also into the summary session, I will try to discuss the topics which you feel you need to repeat, or you have any doubts. So, I will try to repeat those topics in the last classes or summary classes. So, this was today, it was a week 10 live session. Thank you all for joining. I will close the session right now, and if you have any problem, you can just tell me, or I will close the session. Okay, or you can just drop an email to me. This kind of problem solving session we are having on every Tuesday from 6 p.m. to 8 p.m. So, please do join. We have only 2 classes left. So, please join in the last 2 classes and also to the summary classes, because it will help you to summarize or finish the course better. No, the examination, I don't think it won't be objective only. The examination, it will be subjective. Okay, you will have descriptive questions also, not only the objective question. That's why even if I am showing in only MCQs, but I am discussing the topics more thoroughly, so that if there is any descriptive question related to that topic, you can answer it easily. Okay, but there won't be any objective. It will be a mixed kind of questions. Okay, in the final examination. So, thank you all and all the best, and let us meet on next Tuesday in our week 11 live session. I will close the session for today. This class recording and the PPT I will share to you soon. Okay, you can find it on the SWAYAM portal. There is a section known as the problem solving session just below your assignment section. Okay, you can just click on that portion. You will find the Excel sheet. There is the YouTube link for the class recordings, and also you will find the presentation links. Okay. So, now I will close the session for today. Thank you all for joining, and let us meet on next Tuesday from 6 p.m. to 8 p.m.